Hi, everyone. Welcome to the Alliance of Genome Resources office hours. Today, we're going to talk about disease annotations at the Alliance. I'm Yvonne Bradford. I'm a project manager at ZFIN. Co-hosting is Sue Bello.
Hi, I'm Sue Bello. I am a senior scientific curator with Mouse Genome Informatics at the Jackson Lab. Yvonne's going to do the talking, and I'm going to try to handle all the technical stuff. So I'm actually going to turn my video off and mute myself and hand it over to Yvonne. Some technical things. We're asking that you put your questions in the Q&A. And at least for some folks, that Q&A box may be buried under the more. So if you can find that, if you have any questions, and we're going to try to run through the talk and hold any questions for the end because this talk is being recorded and we will make the recording available. So we were thinking it'd be better to hold the questions till the end. All right, thanks everybody. And I will hand it over to Yvonne.
Thanks, Sue. Okay, so if I happen to get any internet problems, I will be turning off my video. I will continue to share though. And then if for some drastic reason something happens with my internet and it goes down, Sue's going to take over. So here we go. Today, we're going to talk about disease annotations at the Alliance. We're going to give you a little bit of background about Alliance disease annotations, how to search for those annotations at the Alliance, how we display disease annotations on the disease page, the gene page, the allele page, how you can download the disease data at the Alliance, and then we'll have a question and answer at the end here. Okay, so first up, background. So the Alliance provides a harmonized data model. The Disease and Phenotype Working Group brought together all of the different approaches for disease annotations across the mods, and we developed a unionized model that accommodates these various approaches. You can get more information about the model if you go to this link. I'm not going to go over the model in detail, but this is a nice representation of what our model is. So this harmonized data model provides us a way of having a unified display of model organism database disease annotations, which the model organism databases use the disease ontology for disease concepts. And then diseases can be connected to genes, alleles, or models. And a model is a representation of the organism, like a strain or a genotype or a fish that is modeling the disease. Okay, so we're going to search for disease annotations at the Alliance. This is the Alliance homepage. On the homepage, there's a text entry box. Oops. It has, you enter your text here. It is a type ahead autocomplete. It provides a list of possible topics. If you hit return after you're done typing, you'll get a full list of the results that match your text input. If you pick something from the list here, that will take you directly to the page that you picked. Okay, so we're going to hit return after having typed in diabetes. And this brings you to this results page. On the left-hand side are categories that are associated with the inputted search text. And the categories here are allele variant, gene, model, high throughput, dataset index, and disease. Here at the top, you can see the full number of results for that input string. And that text is highlighted in the results, showing you why that particular result was returned. And we're going to pick the disease category to do some filtering here. And that produces a new search result for that disease with additional facets for refinement. We're going to look at this a little bit more. So the facets over here, you can refine your results by disease group, and it lists just a high level disease terms, and you can click more, show more to see all of the terms here. The genes that are associated and the associated species. The numbers are counts. As you remember, we clicked disease category to filter, and this just denotes that's what we clicked. Again, the results with the highlighted text. And this one result, we're going to look at that in a little bit more detail. So the name here, diabetes mellitus, if you click on that, that will take you to the disease page. In the results, short definition of the disease is provided. We have some related data topics down here. This is the number of genes, alleles, variants, or models that are associated with this particular disease. If you click this entity, it will produce a new search that is this entity along with the inputted text that you had in the original search, and that leads to a brand new search results page. And I'm not going to go into that in detail. We're going to click on the diabetes mellitus and go to the disease page. So here's a typical disease page. This one is for diabetes mellitus, provides the term name and the disease ontology identifier here in the top left. We have a left navigation bar. As you can see, there are 4 sections on the disease page at the Alliance. We're going to start with the summary section. The summary section provides information about the disease as derived from the disease ontology. So there's a definition, synonyms, cross-references to other disease vocabularies. If you click the show all, you get to see the complete list of cross-references. The parent and child terms are listed and are also links to those particular pages. Again, show all, you can see all the child terms for, in this case, diabetes mellitus. Sources of annotations, so this provides links to the model organism database sources. And we're going to go next to the associated genes section. The associated genes table provides genes that are associated with the disease along with supporting data for that association. I'm going to go into each of these columns in a little bit more detail. Know that you can, all of the tables have a download button at the bottom and you can download the data for that particular table if you would like. All right, so we're going to start with the gene column. The gene column lists the genes that are associated with the disease, the gene symbol. If you click the filter, you can filter by gene symbol. In the gene column, there is also an annotation details link that when you click it provides a pop-up window that provides detailed experimental information to support the gene and disease associations. If you click the name, it'll take you to the corresponding Alliance or model organism database page. A lot of the information that's in this pop-up window is in more detail in the models table, and we're going to go into that more. So just this pop-up, you'll see recurring on various tables. Okay, so the next column is the species column. It lists the species which have direct or indirect data for that particular disease. You can click the species to filter the table. Here we've clicked Danio. You can tell that we've clicked the filter by the shading around the filter, and then the results in the table are filtered by the selected species. So here, Danio. All right, the third column here is the association column. These are association indicate how the gene is associated to the disease. And we have is implicated via orthology, is marker via orthology, is implicated in, is marker for, is not implicated in, or is not marker for. I wanted to note that the not annotations, these indicate that there is evidence that the gene is not implicated or is not a marker for a particular disease, despite expectations that the gene should be associated. Okay. Here, in the associations, we've filtered for is implicated via orthology and is marker via orthology. And just wanted to point out that these gene to disease associations, excuse me, are computationally determined by the Alliance, and they're based on orthology data and disease annotations in orthologous species. And in the based on column for those particular associations, the orthologous gene that the orthology association is based on are listed here. Okay, so then the next column is the disease qualifier column. The qualifier provides additional context for the gene to disease relationship. And those qualifiers are susceptibility to, onset of, severity of, disease progression of, and sexual dimorphism in. And then only qualifiers which have data are presented in the filter list. We have the disease column, which lists the disease terms. In the table also includes child terms. When you click the filter, you can filter by the disease terms. Okay, the next column that we have is the evidence column, and that provides evidence codes that the Alliance uses to justify the disease ontology annotations. If you click on the question mark, it'll provide this pop-up. It will give you more information about the evidence codes. And then we have the based on column, which again provides the orthologous gene that the disease annotation is made with. And then again, clicking the question mark bubble produces a pop-up that explains that a little bit more. And then the last two columns in the associated genes table are the source column and the references column. The source column provides the database that is the source of the disease annotation, and the references column provides the supporting references for that particular row of data. Okay, and so that's everything for the associated genes table. We're going to jump next to the associated alleles table. The associated alleles table provides allele associations with disease along with supporting data. The filters here behave the same way they do as the gene table, so we're not going to go, they're essentially the same and we're not really going to go over them. I'll just note that for the alleles, there's only two associations that are relevant for the allele to disease association, and that's implicated in and is not implicated in. Again, clicking the annotation details brings that pop-up window for detailed experimental information for the allele disease associations. And then the last table on the disease page is the associated models table. This table provides an organism level view of models of human disease with the supporting data. Again, the filters behave as the other filters on this page behave. This table is quite large. There's a scroll bar that indicates there's more data available in the table, as well as gray shading that indicates there's more data behind the fold. We're going to start here with the models column. So a model is a strain, a genotype, or a fish. It is a representation of the organism that has been shown to model a disease. The name here, if you click on the name, it takes you to the corresponding model organism database page. Again, we have the annotation details pop up that we've seen in previous tables. A new table in the associated, or a new column in the associated models table is the experimental conditions column. This provides information about conditions that have been applied to the model. This data is annotated for zebrafish and C. elegans models, and here you can see we've filtered the table for species, Danio, Rario, and C. elegans. Another new column that we have here is the conditions modifier column. This provides information about conditions that have been applied to modify the model. And when we say modify, we're talking about amelioration or exacerbation of the model. And this data is annotated for C. elegans models. And the last new column that we have in the associated models table is the genetic modifier column. And this provides information about genome features that are applied to modify the model. And this data is annotated for C. elegans and Drosophila. So you can apply various, you can apply multiple filters to the table at one time. Um, it- and you can then download that information. So any filters that you apply uh, in the table and then you click download, those filters are applied to the data that's downloaded. And then this is just a kind of a sample of what is downloaded. The data is in a TSV format, and it includes all of the identifiers for that particular data set. All right, so now we want to move from the disease page to the gene page to uh, see the disease annotation, disease data displayed there. So how do we get to the gene page? From the disease page, we can go to the associated genes table, and in the gene column, you can click on one of the gene symbols, and that will take you directly to the Alliance gene page. You can also go to the search box and type in the gene that you're interested in and then pick from the pick list to go directly to that gene page. So here we're going to pick CFTR for zebrafish gene. So here's the Alliance gene page. Again, we have the left navigation bar. In the upper left-hand corner, you can see the gene symbol, the species, and the database identifier for that gene. Um, and we're going to go to the disease association section to see the disease associations for this gene. So the disease association section on the gene page has three main sections or areas. We have an area to select orthologs to be included in the ribbon and the table. The ribbon is a visual representation of the disease associations that are disease annotations that are associated with the gene and its orthologs. And then the table is just detailed annotation information for the annotations that are represented in the ribbon. So we're going to kind of look at the orthology section here first. So you can pick orthologs to display in the ribbon and the table. If you click on the stringency button, you get a drop-down menu where you can pick the stringency. You can pick how stringent the filter is that you would like to apply. By default, stringent is what is picked. If you click on the question bubble, this pop-up is produced that gives more information about the various stringencies that are available. You can click to include negative annotations in the table. By default, this is not clicked. Again, negative annotations are cases where the expected disease association was not found. If you click the species button, this drop-down menu is provided and allows you to pick which species that you would like to display. By default, all of the species are displayed. If you have a species in the list that is grayed out, that means that species has no data. Okay, we're going to now move on to the ribbon section. The ribbon is a visual representation of disease annotations that are associated with the gene and its orthologs. Um, the gene of interest or the gene page that you're on is always listed first in the ribbon. The highlighted boxes indicate that that particular column has been selected to display data in the table. The colored cells in the grid denote the presence of disease annotations for that particular topic and gene. And then the darker the colors, the more annotations there are for that particular uh, category and gene. So the ribbon categories are derived from a subset of gene ontology terms identified by the Alliance as good high-level grouping terms to group disease annotations. So our groups are disease by infectious agent, diseases of anatomical entity organized at the system level, diseases of cellular proliferation, genetic diseases, and then all other diseases. Now we're going to move on to the table that is the uh, more detailed information for the ribbon. That again, the gene whose page you are on is displayed first in the table. This table is very similar to the other disease tables that we've already seen. The columns and filters are the same as those that we've seen on the disease page tables. If you click on a gene symbol, you'll go to the Alliance gene page. I want to point out that the source column provides the source of the annotation. Sometimes you'll see OMIM via RGD as a source. This indicates that the annotation originated with OMIM and that RGD mapped that OMIM term to disease ontology term and then added the evidence code for that annotation. Okay, so um, we can also see disease annotations on the allele page. And to get to the allele page from the gene page, we are going to go to the left navigation bar and click the alleles and variants section, and that'll take us to the allele and variants area. In the table allele and variants, the symbol, you can click on that. That links to the allele page. In that same table, you can tell if a particular allele has a disease annotation looking at the has disease annotations column. Yes means it does have disease annotations. If there's nothing there, it means it does not. We're going to click the yes. And that takes us to the allele page in the disease association section. Again, the allele page has the familiar left navigation bar with the allele symbol, database identifier, and species identified. In the disease associations table, it lists the diseases which that particular allele is associated with. Um, we have a column for annotation details. You can click that view button and it will produce the same pop-up that we've seen in the other tables to provide the detailed evidence for that association. Okay, now we want to download disease data from the Alliance. To do that, uh, you can go to the Alliance homepage, you click on the data and tools, go to the downloads page. That takes you to this page, which has downloads for various data types. We're going to focus on disease. It lists, you'll see a list here of various files that we can download. All disease annotations is this first line here, and then you can also download by, if you were interested in a particular disease species, disease annotations, you can select one of those files as well. Files are provided in JSON and TSV format. And I think that wraps it up. I want to thank you for your time. And if there are questions, I would be happy to take those. Sue and I would be happy to take those. Thank you.
Thanks, Yvonne. That's great. So do we have any questions? Oh, and you want to go to the next slide and plug the next office hours?
I absolutely do. We would like to invite you to the next Alliance office hours. That is going to happen April 17th, 2025 at noon, where Kimberly Van Auken will present on how AI informs literature curation. You can register if you go to the Alliance homepage, click on news, go to the event calendar. You can register there. You can also select to watch recordings of previous office hours webinars. So we hope that you can join us at future office hours.
Okay, well, if there aren't any questions, then I will thank everyone for coming and hope to see you again at the next office hours. I will also say that there are, if you think of questions later, the help will get you to our user community where you can always ask. And somebody is always happy to respond. Okay. Thanks
Thank
you,
you.
everyone. I'm going to
Thank you.
stop the recording.
Thank you.